everybody. Hope you had a good mini break. Let's see. We're going to pick up where we left off before break. There's a problem set that is hard copy there. I'll put it up. I'll put the Word document up on Moodle. You can do it either way. It's using the equations to learn more about respiratory physiology. It's due on Sunday night at the CST desk. I'm happy to answer questions about it, anytime between now and Sunday night. Let's see. So interesting bit of news this week. The Nobel Prize in Medicine was awarded to 3 scientists for oxygen sensing in cells. So we're obviously thinking about oxygen today, and so this year's Nobel Prize is actually relevant. I mean, it's not about respiration per se, but it's about how cells detect oxygen and change gene expression as a result of either high or low partial pressures of oxygen. It's pretty interesting. In cell-phys, they're doing glycolysis and aerobic respiration, and here we are doing, human respiration, and it's Nobel Prize season. So what you're learning about is Nobel Prize winning. Okay, so today what we're going to do is focus on the Davenport diagram, make sense of that thing that I revealed a week ago, but try to have it make sense. It's complicated. And then we'll talk about how the blood transports oxygen from the lungs to the tissues and transports carbon dioxide from the tissues back to the lungs. So here's the Davenport diagram that I showed on a week ago. It's a graphic representation of the Henderson-Hasselbalch equation. So remember that the Henderson-Hasselbalch equation comes from the fact that CO2 and water form carbonic acid, and that it dissociates, like all acids, into strong acid and a weak base. And this is just the Henderson-Hasselbalch version of this acid equilibrium. Those of you who have this in biochemistry, I don't need to go into it anymore. It's just the straight up Henderson-Hasselbalch equation. But what we do in respiratory physiology is convert it into this graphic version. So let's see if we can make, so I will not ask you to use the equation. I won't ask you to memorize or use this equation, but I will ask you to know how to use and interpret a Davenport diagram graphic version of it. So let's, and here are some other equations that, well, here are the ventilation equations that will come in, that you'll need for the problem set. So let's work on this and try to make sense of it. And I've discovered that the Davenport diagram has all these lines all over it, and it's hard to interpret when it's all there. So what I usually do, what works best, I think, is to build it. So that's what I'm going to do here. I'm going to build the Davenport diagram. So in the Davenport diagram, the x-axis is plasma pH. The pH of blood plasma. And the y-axis is plasma concentration of bicarbonate ion. So the x-axis is pH. So this, the Henderson-Hasselbalch, you would ordinarily think that this should be the y-axis and this should be the x-axis, the way the Henderson-Hasselbalch equation is written. But the Davenport diagram reverses it for some reason. So pH is on the x-axis and bicarbonate is on the y-axis, always. But it's still just a representation of this equation. A variable, it's actually a three-dimensional equation because this is variable as well. So what we do in order to make it two-dimensional is to plot bicarbonate as a function of pH for a given arterial CO2. So we're going to start with just a fixed arterial CO2. And let's start to put up some normal human values. Normal human value for the plasma pH is 7.4. A normal human value for the plasma bicarbonate is 26. So this is in millimolar units, and this is, in pH units. It's unitless. So the normal value for respiration, bless you, what we're doing, all of us sitting in here are at very close to pH 7.4 in our blood plasma, and our blood bicarbonate concentration is very close to 26 millimolar. We operate best in a very narrow range of blood pH. If our blood pH, I'm sort of arbitrarily drawing a normal range, from something like 7.3 to 7.5. If we go outside that range, things start to fall apart pretty fast. By fall apart, I mean the enzymes that we need to survive don't function well. They have evolved to function at pH 7.4 plus or minus a fraction of a pH unit. So that's a sort of a representation of the normal range. Let's just put 7.2 down here and 7.6 up here, just to give you a sense of the scale. Let me just see if I did that right. Yeah, pretty close. We are at a given PCO2. That's what I said. So this is a plot of bicarbonate as a function of pH. It's a plot of bicarbonate as a function of pH at a given PCO2. And so what that means is that at a given PCO2, the Henderson doesn't do that. It does something like that. If we were to plot the Henderson-Hasselbalch equation on a linear graph, because it's got logarithms, there's some curvature in it, it would look something like that. Oh, by the way, just to be clear about the range, so this is maybe 20 millimolar, and this is, so that's 6, so this would be 30 millimolar. Right? That's the range of bicarbonate concentrations on the y-axis. So this black curve represents the Henderson-Hasselbalch equation at PaCO2 equal to what's normal? 40. 40 millimeters of mercury. I will tend to be a stickler about units, so make sure if you've got a term that has units, you include units. When the problem said it, it's required. So the units for PaC, arterial CO2 is millimeters of mercury. The units for ventilation is in mils per minute. And the units for production of CO2 is in milliliters per minute. Okay, so this is the curve that we are working on in terms of respiration when we have an arterial CO2 of arterial partial pressure of CO2 of 40 millimeters of mercury. So this number then becomes a number, this number becomes a number, and now we have a simple equation that relates bicarbonate with pH. And that equation defines that curve. Okay, everybody with me? And as long as our arterial PCO2 is 40, We have to be on that curve. It's defined by acid-base chemistry. Our blood plasma, has to be on that curve, as defined by acid-base chemistry. So for example, if something in our body produces acid, and we don't change respiration, which direction would we move on this curve? Something is producing acid, we would acidify. pH goes down when there's acid. And so that, let me use a different color here. That would be an acidification that was not due to any change in respiration. That we're still on the PCO2 curve of 40, but we have added acid to our system. And the example I think someone came up with on Tuesday, or last week, was exercise, producing lactic acid, adding acid to the body. And so this is called metabolic acidosis. Metabolic in this room for today just means not respiratory. Anything that produces excess acid and doesn't change a respiration, that is to say we're still on the PCO2 curve of 40 millimeters of mercury, is metabolic. And this would be metabolic acidosis. And so you can imagine what is this? What do we call that? Metabolic alkalosis. What could cause us to lose acid in our body? And it's not respiration. So this is metabolic alkalosis. Remember, this is our starting point, our normal point. Kendra, what could cause us to have excess alkaline or insufficient acid?
Is it because the, like, going off of the lactic acid thing, is it because the lactic acid gets too slow?
No, no, not really, because if we produce lactic acid and then we stop producing it, it's going to rebound back to normal. It's not going to overshoot, typically. Well, so one way we can accumulate too much alkaline is kidney disease. If the kidneys are not excreting, so I'm trying to bring in just some ideas. If the kidneys are not excreting alkaline enough, then we're going to accumulate alkaline, and that would be metabolic alkalosis. Another is, we're going to get into the gastrointestinal just for a moment here. Vomiting. Chronic vomiting for some reason. You know that stomach acid is acid. Chronic vomiting would be the excretion of excess acid. That can produce metabolic alkalosis because you're excreting excess acid due to chronic vomiting. Okay? The point is, when we're on this curve, respiration, we've said respiration is normal. Okay, so now, but you know respiration isn't always normal. Let's go to a different curve. Let's go to PaCO2 of, let's say, 20. I'm going to go extreme. This might actually not be survivable, but just to be extreme. PaCO2 of 20 millimeters of mercury. it's a curve. It's basically a curve that's parallel to the initial curve. It's the Henderson-Hasselbalch equation, plugging in 20 here instead of 40. It's the same equation, everything's the same, except that now we're at a PaCO2 of 20. Is this hyperventilation or hypoventilation relative to normal? This is hyperventilation, because we are ventilating out more CO2 than we need to. And PCO2, this has gone up, PCO2 has gone down, hyperventilation. Normal production of CO2, excess ventilation, hyperventilation, low PCO2. So this is hyperventilation. On the red curve. So if we move, so basically this arrow, and we don't move X and Y, we move for some reason that I actually don't completely understand. It's got something to do with buffers. It's got something to do with innate buffering. When you move from normal ventilation to hyperventilation, you move along an axis that looks like that. So if I were to ask you to hyperventilate, which you could do because you have control over your ventilation to a certain point, I could ask you to have hyperventilate, and you would move from this curve. Now remember, there's a whole family of curves, right? PCO2 of 39 is here, 38, 37, 36, 35, 34, all the way to 20, right? This is a two-dimensional surface, but we're just drawing one line here at 20. PCO2 of 30 would be, in between. Clear? I'm just using this one example. So if I were to force you to hyperventilate, you would move along this line until you got to whatever PCO2 you had. And so this red line is called respiratory. Respiratory. Is it acidosis or alkalosis? Alkalosis. Respiratory alkalosis. Your body has not done anything yet, but you've changed your respiration. So you've moved from the normal point to a new curve, a new respiratory curve. Now, what would be the body's response to hyperventilation? Well, does it like being up here at 7.45 or whatever it is? No, it needs to be closer to 7.4. The way I've drawn it, it's still within the normal range, so the response is not going to be extreme, but 7.4 is the set point. And so the body is going to respond by moving down this curve so that it's back to 7.4. We're hyperventilating, so we have to stay on this curve as long as we're hyperventilating. But the body can compensate for that hyperventilation by retaining acid. And so this one right here would be called metabolic compensation for respiratory alkalosis. So this is metabolic compensation. It's not metabolic acidosis because it's using the curve to try to normalize, not to go acid, but to go back towards 7.4. That's why it's compensation. But it's metabolic, because the respiration is on this curve, as long as we're hyperventilating. Now the body is compensating to try to bring the pH back to 7.4. Okay, so let's do another example. Let's do green. Okay, so here's if we hypoventilate, PaCO2 equal to 60, say. Here's number 3. We're on the curve. We're on the Henderson-Hasselbalch equation, plugging 60 in here. If this is hypoventilation, PCO2 is too high, ventilation is low, PCO2 is high. So this movement here, if I ask you to hold your breath, to hypoventilate, your body would respond by moving along that arrow. The same buffer curve, but moving towards hypoventilation. And again, you can see that we start to get outside of the desired range. And so what is going to be the body's response to hypoventilation? It's going to try to excrete acid to bring it back to a normal value of pH. The body doesn't really care about the bicarbonate concentration. We can go up that high. It cares more, the control point is set as pH, not bicarbonate concentration. The body can tolerate a wide range of bicarbonate concentrations, but it cannot tolerate a wide range of pH. So what do we call this green line? Use your pattern recognition skills to tell me what we call this green line. Respiratory acidosis. And I'm not going to label it because you can see why. But you should probably label it. This green line is respiratory acidosis. What is this green wiggle line? Metabolic compensation for respiratory acidosis. So the point is, for each respiratory condition, hypoventilation, normal ventilation, or hyperventilation, we're on this curve as defined by the Henderson-Hasselbalch equation. We're on one of these curves, and so when you change respiration, you move from one curve to the next. When the body, if the body changes something, you have to stay on that curve of, if the body changes something and you're on the normal ventilation, it's acidosis or alkalosis. If you have a respiratory defect, the body can compensate for it. If you have a respiratory defect, the body tries to compensate for it. What might cause, well, no, I'll leave that alone. You know what might cause hypoventilation, asking you to hold your breath. Lung disease. You know what might cause hyperventilation. Anxiety attack, a panic attack can cause hyperventilation. Other things. So now you can see, so it got complicated and it got messy pretty fast. But so when I show you this, I missed a couple of arrows. Okay, so let me turn that off and make it right. Okay. So let's say we went to metabolic acidosis. Lactic acid, kidney disease, metabolic acidosis. Your body can compensate to metabolic acidosis by I should use blue. So you have a disease, kidney disease, let's just say kidney disease, metabolic acidosis. Your body can compensate by ventilating more. Now you can see in the way I've drawn this arrow, I'm not getting all the way to 374, but I'm going the right direction. So what is this blue, what do we call this blue arrow? If this is metabolic acidosis, what do we call that one? Respiratory compensation for metabolic acidosis. And let's go to metabolic alkalosis, kidney disease or excess vomiting. Your body can compensate, so say we go up to here, your body can compensate by hypoventilation. It's not hypo now, it's respond by slowing ventilation. So what do we call this arrow? So this is metabolic alkalosis, and this would be respiratory compensation for metabolic alkalosis. So this arrow here is respiratory compensation for metabolic alkalosis, just the way this arrow here is respiratory compensation for metabolic acidosis. So the point is, if you are in a situation of metabolic alkalosis or metabolic acidosis, the respiratory system can change to compensate, to help improve the pH situation. Just the way if respiration is abnormal, the body can compensate to try to bring the pH back to normal. pH is the important variable. So now you can see why there are certain arrows that aren't drawn. Would the body ever do this? Go from metabolic acidosis to this? No, that's making it worse. That would never be a compensation. That would be a combination of a body acidosis and respiratory acidosis. That would be two defects happening on top of each other. So that's not compensation, that's two defects. And we can get to that point in two ways. I mean, you can see, we can have metabolic acidosis and then respiratory acidosis, or we can have respiratory acidosis and metabolic acidosis, right? The main point is that respiration defines which of these curves you're on. But the body can put you in various places along that curve, the body being the kidneys, the muscles, the digestive system. Okay, so now let's put up, so I don't know, if the best way might be to draw 2 or 3 Davenport diagrams to have, in order to keep the arrows straight. But people don't do that. People just draw one Davenport diagram and label it. So now, so here's the Davenport diagram. We start, the normal point is at a pH of 7.4 and a bicarbonate concentration of 26 or so. metabolic alkalosis, metabolic acidosis. But because we're at a normal PCO2. Respiratory compensation for the metabolic alkalosis is trying to normalize the pH. Metabolic compensation for respiratory acidosis is trying to normalize the pH. So this slide tries to differentiate between metabolic alkalosis and focusing on the kidney. We haven't learned about the kidney yet. We're going to learn about the kidney next, but you can see why the kidney and the respiratory system work together to control blood pH. So if you say renal compensation for this effect, I'll accept it. But for now, we're just calling it metabolic. Metabolic being anything besides respiratory. So we have used the Henderson-Hasselbalch equation, but in a highly graphical way. Questions? So my recommendation for learning this is to practice drawing it from scratch, thinking about what is normal PCO2. Are we talking about hyperventilation, which is respiratory? Hyperventilation is respiratory alkalosis, and then what would be the body's response? Or then, perturb it. Let's do metabolic alkalosis. What would be the body's response? That would be hype to ventilate less. So I don't know. It doesn't come naturally, but it makes a lot of sense when you learn how to use it. By the way, this graph uses the expression milliequivalents per liter. Those of you in chemistry know that a milliequivalent is the concentration of charge, and bicarbonate is monovalent. So milliequivalents per liter is, for all practical purposes, the same as millimoles per liter, which is millimolar. If we were to do this at advanced A&P, I would discriminate between milliequivalents and millimoles. We don't have to do that. Giving you time to ask questions. Or maybe the best thing, okay, so let's talk about how these things can change, right? Respiratory acidosis. So what am I doing? I'm looking at respiratory acidosis. Hypoventilation. High CO2. So respiratory acidosis, hypoventilation, an increase in PCO2, dyspnea, which means not breathing right. This means abnormal. Pnea is referring to respiration. Asthma, not breathing right. Emphysema, lungs not doing what they need to do. Respiratory alkalosis. Respiratory alkalosis is hyperventilation. Panic attack. Altitude. We'll talk about altitude in a minute. How many of you have ever been to something like 10 or 12 or 14,000 feet? Did you notice anything about your respiration? Did you feel like you were panting? Just sitting, drinking a beer? Not drinking a, whatever it was.
Yep.
You can hyperventilate when you go to altitude. We'll talk about that. Metabolic alkalosis. Metabolic alkalosis. Metabolic alkalosis. This defect here, we've talked a little bit about. Metabolic alkalosis down here, vomiting, for example. Metabolic acidosis, lactic acid production by anaerobic metabolism, kidney failure, diarrhea. Loss of fluids by diarrhea can cause metabolic acidosis. So people with cholera who are losing fluids by chronic severe diarrhea actually have metabolic acidosis going on at the same time. Let's do some clickers and just see how well this is sunk in. Nice. What happens to blood pH if you go to high elevations? Look at the Davenport diagram. What happens to blood pH if you go to high ventilations? Okay, good. I'm seeing a lot of correct answers. It increases. Respiratory alkalosis. Why? So we're going to talk on Thursday about the control of ventilation. But basically, at this level, the thing to think about, if you go to high elevations, like at 15,000 feet, you're breathing about half as much oxygen. 20,000 feet, it's half as much PO2 in the atmosphere as at sea level. So we are acclimated for sea level. You go to high elevations, PO2 decreases because pressure decreases. You're at high elevation. Your body responds to the decreased oxygen by saying, I need to breathe more. I need more oxygen. But as far as CO2 is concerned, remember at high elevation, 0 is 0, right? I mean, the PCO2 constant, the partial pressure of CO2 at elevation, is insignificantly different from the partial pressure of CO2 at sea level. So when you hyperventilate because of oxygen, you're hyperventilating when you're trying to normalize your oxygen by ventilating more. You're actually hyperventilating with regard to CO2. And CO2 is the important thing for pH. So going to high elevations and trying to normalize your O2 causes you to hyperventilate as far as CO2 is concerned. Okay, what happens to blood pH during an anxiety attack? During an anxiety attack. The anxiety attack is going on. Yeah, I'm seeing a lot of correct answers. It increases. It's hyperventilation. It's the same thing. Hyperventilation. This isn't going to altitude. This is happening at sea level during a panic attack. Okay, what happens to blood pH during exercise? Those of you who chose A and C are correct. It decreases a little bit. It stays the same or decreases a little bit. Exercise, lactic acid. We don't produce a lot of lactic acid, but we produce a little lactic acid. So pH decreases due to lactic acid production. That's getting it. Okay, one of the cures for hiccups is to breathe into a paper bag, to rebreathe your exhaled CO2. What happens to blood pH? It actually doesn't work, but people do it. What happens, it doesn't work for the treatment of hiccups, that is. What happens to blood pH? Yes, it decreases. Why? Because when you rebreathe CO2, you're breathing now an elevated concentration of CO2, more CO2, more acid, decreased pH. Okay, good. I don't know if you have more. Okay, yeah, so that's it for the clippers. but again, the clickers aren't for testing, the clickers are for answering questions. Are there any questions? Okay, so there's a figure in your textbook that describes the matching of ventilation and blood flow. And the book makes a big deal out of it, but we're not going to make as big a deal out of it. But I do want to mention this. The idea is, let's just look at this flow chart. Decreased air flow. Okay, what would we call that? Is that respiratory or metabolic? It's respiratory. Decreased airflow would be a respiratory effect. Decreased airflow would that be hyperventilation or hypoventilation? Hypoventilation. Okay, so now we're not talking about CO2, we're talking about oxygen. And the point is, if there is hypoventilation, the blood vessels surrounding the alveoli in that section of lung that is hypoventilated constrict. And so the blood flow there's an attempt to match blood flow with airflow. Why would we want to, we call airflow ventilation, as I've said before, and we call blood flow perfusion. The blood is perfusing the alveoli. The alveoli are ventilated with air. There's a matching of perfusion with ventilation. If there's an area of lungs that is getting decreased ventilation, we would want to divert the blood to perfuse the areas of lungs that were not getting decreased airflow, that were getting normal airflow, and that's what this is all about. By decreasing blood flow to that part of the lung, the blood could go elsewhere, where it would be better matched for ventilation and perfusion. This property of detecting oxygen is what the Nobel Prize of this year is all about. The cell's ability to detect oxygen and to respond. What they really came up with, I mean, the reason why they got the Nobel Prize was decreased blood oxygen, hypoventilation, is associated with the production of erythropoietin, which is a protein that helps in the production of red blood cells. Some of you may have heard about athletes taking EPO, EPO. EPO is erythropoietin. It's a hormone that increases the production of red blood cells. So athletes, it's illegal, but if erythropoietin is very helpful for the people who have chronic hypoventilation, say with asthma or emphysema, because if we can increase the blood's ability to carry oxygen, we can help mitigate the effects of hypoventilation. But it's used by athletes to increase their body's ability to carry oxygen by increasing their red blood cell production, but it's against the rules. And those of you who are athletes don't do it. Anyway, this point here, detecting PO2, is this year's Nobel Prize in Medicine. Okay, let's talk about a region, a different defect. Decreased blood flow to a region of the lung. Let's say some of the pulmonary capillaries have gotten blocked, or pulmonary arterioles. Can that happen? Sure. A clot, pulmonary embolism. You know, there's lots of reasons why blood flow might not be normal. Well, if there's low blood flow to a region of the lungs, those alveoli are getting insufficiently perfused with blood. So their CO2 is going down. They're getting ventilated, but they're not getting perfused. So that would lower PCO2, which causes constriction, not of the blood vessels, because they're already got a problem, of the bronchioles. In other words, we can constrict bronchioles to match an area that has decreased blood flow, so that the air goes to the parts of the lungs that are getting fully perfused. The bottom line, I'm making more of a big deal than I intended to. The bottom line is that there is matching locally in small parts of the lungs between ventilation and perfusion. What do you suppose causes, let's just make sure we understand, what causes vasoconstriction, or what causes bronchoconstriction? What cell type are we talking about? Smooth muscle. Arteriolar smooth muscle, bronchiolar smooth muscle. Is calcium important in this story? Yes. Q, calcium, cross-bridge cycling, yada, yada, yada. I could ask you about cross-bridge cycling again because I asked, I could ask you what's the molecular mechanism of the vasoconstriction that allows for ventilation, perfusion, matching. I'm not going to do it again, but you should do it again. Because constriction of smooth muscle is what does this function of the lung. Questions? Okay, so now this is an equation that I will not ask you to learn for a test, but we're going to use this in lab. So I want you to have this equation sort of at your fingertips for lab on Thursday. It's an equation that, let's just unpack it. This is alveolar partial pressure of oxygen. This is the partial pressure of oxygen in inspired, that's what the I stands for, inspired air. And it's typically about 150, because remember it's not 160, it's 150, because what gets into the alveoli has been humidified. That's the inspired part. Here's the partial pressure of CO2 in the alveoli. And now here's an interesting number here. So the respiratory quotient is what this is called. It's the production of CO2 divided by the requirement for oxygen at a given body condition. So this is the production of CO2 and this is the consumption of oxygen. And what's interesting is it depends on the diet. Isn't that weird? So if you eat pure carbohydrates, you eat nothing but potatoes, white bread, your respiratory quotient is 1.0. I can make this make sense. If you eat nothing but carbohydrate, I'm going to do this over here for the purpose of the camera. So remember, let's do some self-is. Still relevant. 36H12O6 plus O2 goes to, let's do glycolysis, let's do all of aerobic respiration. CO2 plus H2O. That's complete combustion, complete metabolism of carbohydrate, a simple carbohydrate, a sugar, glucose. Now we got to put in some numbers. See, I haven't done this. So we got 6 CO2s coming out because we have 6 carbons on the side, so that one's easy. How much water do we produce? 6. Because we have 12 hydrogens, and we need 12 hydrogens there, so that's that. So how much oxygen do we need here to balance this equation? So we have 12 plus 6 is 18 oxygens. So we need 18 oxygens, 6 plus 12, so that would be 6. Does that work? So there's the complete combustion of a carbohydrate. 6, 6. The ratio, the quotient is 1. If you eat a pure protein, well, let's go to fat. Let's just, it's kind of gross to even talk about, but if you were to eat a pure fat diet, eat nothing but butter. a good source of calories, but it's a pretty bad source of nutrition. If you do the same equation for fat, which we didn't do in self-phys, and I'm not going to do now, but you can live on fat for a while, and if you do the same equation, you find that the respiratory quotient is 0.7. It produces less CO2 for the amount of O2 that it consumes. And if you eat a diet of pure protein, it's about, it depends on the protein because proteins are not all the same. Protein is about 0.85. And if you eat a mixed diet, which we all typically do, of carbohydrate, protein, and fat, the respiratory quotient is typically 0.8, somewhere between fat and pure fat and pure carbohydrate. So this equation, which we'll use in lab, defines the alveolar O2 as a function of what you eat and what you breathe in terms of your ventilation of CO2 and what you breathe in. So we'll use this equation in lab. Let's take a break and we'll talk about the transportation of oxygen by the blood. Let's take a break. Name hemoglobin. Hemoglobin is a tetramer of similar protein subunits, globin, and each globin monomer has a heme group attached to it. That makes it hemoglobin. And so here is the structure of heme. So here's the heme group. Here it's got iron in the middle of it. This is why iron is an essential nutrient. You have to eat a certain amount of iron in order to make heme. The heme group is covalently attached to the globin, right there. This is all of the protein, all of this protein. And oxygen binds to this iron-nitrogen complex here. So there's high affinity and actually cooperative binding of oxygen to heme. So this is a curve. So this is an important curve. This is plotting the amount of hemoglobin, the percent of hemoglobin that is saturated as a function of the arterial, well, as a function of the partial pressure of oxygen. This is the hemoglobin binding curve. Now, you may be thinking, okay, this is a linear scale. This is a linear scale. This is also a linear scale. You might remember from cell physiology that when we did a linear scale of ligand, in this case oxygen, and the amount bound, we got a curve that looked something like that. This looks a little different than that. It's got this S-shaped bit down here because of cooperativity, because binding of oxygen to one of the myoglobin monomers increases the affinity of the binding to others. It's a cooperative allosteric interaction. Binding of oxygen to this, let's just say this myoglobin, this heme group of this monomer, increases the affinity of binding to its neighbors by a conformational change in allosteric mechanisms. And so, how many of you have done cooperativity in biochemistry? Okay, so you've seen how you can turn a curve that looks like this into a curve Have you seen how you can turn a curve that looks like this into a curve that looks like this with cooperativity? Anyway, that's what happens. And we're not going to get into the chemistry of cooperativity. I'm just pointing out that hemoglobin has a cooperative binding effect, and that's reflected here. So now, some oxygen is just dissolved in plasma. Right? Oxygen dissolves in plasma just like pretty much any other gas. But very little of the blood, of the oxygen that is being carried by your blood, is being carried as dissolved oxygen. 90-something% of it is carried as bound oxygen, bound to hemoglobin. So this hemoglobin binding curve is really important because that's what allows us to carry enough oxygen. If we depended on just carrying oxygen dissolved, we wouldn't be cities. So at a PO2, arterial PO2 is 100 millimeters of mercury, and so normally at a systemic arterial PO2, we are 98% saturated. 98% of your hemoglobin is saturated with oxygen. Hemoglobin is 98% saturated in the normal arterial blood. Normal venous blood is around 40 millimeters of mercury. Now this is not the same 40 as the PCO2, right? We're talking about O2, but venous blood has a PO2 low, right? After your body has consumed oxygen, the venous blood, bluish in color, has a low PO2, and so, but even at low PO2, we're still at something like 70% of the hemoglobin is saturated, is bound with oxygen. So this is the range in which we normally work. Arterial, venous. We're working in this range from 98% saturation to 75% saturation. That's the range. So even venous blood, after it's been drained of as much oxygen as the body takes from it, is still 75% saturated with oxygen. It's just an interesting fact. Let's talk about how your body carries oxygen. Let's talk this one out. So here is a cartoon from your textbook that describes the movement of oxygen in the lung capillary and the movement of oxygen in the tissue capillary. Notice here we have an alveolus, and here we have cells that are consuming oxygen. So this is in the lung, here's in the tissues. Oxygen is being moved, but each of these arrows represents a word. Let's see what is the word that defines getting oxygen into alveoli? There's one word. Air pressure. If we just, there's air pressure here, but what do we need to get the air into our lungs? One word, begins with V. Ventilation. This is ventilation. Right? What were some of the other words that you were coming up with? I mean, we need air pressure, certainly, but if we just have air pressure and we don't ventilate, the oxygen doesn't get into our lungs. You might be thinking negative intrapleural pressure. Yeah, sure, that's necessary for ventilation. Okay, maybe I won't play this quiz game because it's like trying to read my mind, but this arrow here is ventilation. Okay, what is the mechanism by which oxygen gets from the alveoli into the blood? Remember, we've got type 1 alveolar cells, and we have endothelial cells, and we have this very thin set of cell membranes. What word defines this arrow? Diffusion. So we have ventilated and diffused oxygen to get it to be now dissolved in the plasma. But remember, the plasma doesn't carry very much. It carries less than 2 of the total oxygen that is dissolved. What is this arrow? What does this arrow represent? Moving from blood plasma into the cytoplasm of a red blood cell, an erythrocyte. What mechanisms bring oxygen from serum into red blood cells? It's diffusion. We have no transporters for oxygen, so it's got to be by diffusion. What does this arrow? So now we have moved dissolved oxygen in the plasma to dissolved oxygen in the cytoplasm of a red blood cell. What does this arrow represent? We take hemoglobin, and now we have hemoglobin with oxygen on it. What's the word for that reaction? Binding. That's binding. Okay, oxygen is easy. We ventilate it, it diffuses, and then we bind it to hemoglobin. Pretty easy. And now, what is the name of the process that brings this red blood cell from the lungs to the tissue? Blood flow. I mean, blood, right? Blood flows from the pulmonary capillaries to the tissue capillaries. And I could ask you, okay, what are the anatomical structures that it moves through, right? I could ask you again to tell me about, venules and the pulmonary veins going into the left atrium, going through the bicuspid valve into the left ventricle, getting pumped from the left ventricle into the aorta and into some artery, and then into arterioles, and then into capillaries. That's some tissue. I've already tested you on that. That doesn't mean I couldn't do it again. Okay, so now we have moved red blood cell from the lungs to the tissues. The tissues need oxygens, and they're consuming oxygen. So what is the name of this arrow? What are these arrows representing? All of these. Diffusion. Diffusion from the cytosol of the red blood cell into the plasma. Diffusion from plasma into extracellular fluid. Diffusion from extracellular fluid into the cytoplasm and then into the mitochondrial matrix, which I hope you remember from cell physiology, is where oxygen is required as the final electron acceptor in the electron transport chain for the oxidative production of ATP. That's what we're consuming oxygen. We're reducing oxygen to water in the mitochondrial matrix. So oxygen is simply diffusing down its concentration gradient because it's getting consumed here, and then it's diffusing in response to that consumption. What is the name of the process that defines this arrow? Unbinding, or dissociation. Oxygen's pretty easy. There are some factors that, so now we're talking, I want to talk just for a moment about the factors that alter binding and unbinding of oxygen to hemoglobin. There's a couple of graphs, but bottom line, heat, so look what happens. So here is the hemoglobin binding curve at normal temperature. Here's the hemoglobin binding temperature at a cold temperature, and here's the hemoglobin binding temperature at a high temperature. Watch what happens when we go from normal temperature to high temperature. Is binding increased or decreased? It's decreased. Binding is decreased. We let go of this much oxygen from the hemoglobin when we go from normal temperature to high temperatures. This actually we use when you are exercising. You produce heat, you know that. You start to sweat in order to compensate for the increased production of heat. If you didn't sweat, you would become hyperthermic, you overheat, and so you produce excess heat when you exercise. So your cells, the muscles, are producing heat, and that assists in the that heat assists unbinding, unloading of oxygen from hemoglobin in the tissues that are hot. I don't think your muscles get all the way to 41 degrees, but elevated temperature assists the unbinding of oxygen from hemoglobin. And it's not the only thing that does it. Here, let's look at this one. Here's normal PCO2. Here is, oh, sorry, this is all CO2. Here's normal pH, and here the yellow is acid pH. When we move from here to here, acid assists in the unloading of oxygen from hemoglobin. Lactic acid produced by exercising muscles would assist in getting that oxygen off of hemoglobin and into the tissues where it's needed by the mitochondria. At a given pH, high CO2 assists in the unloading of oxygen. So this is all of these things, temperature, pH, and CO2, all assist in the unbinding of oxygen from hemoglobin. And we use that physiologically. So now that we've just talked about how high CO2 can assist in the unbinding of oxygen, let's talk about how your body carries CO2. Okay, this will be the last graph of the day. We're going to begin in the tissues, metabolically active tissues, brain, muscle, consuming oxygen, producing CO2. Self-is question. What processes in aerobic respiration produce CO2? Two good answers.
Citric acid cycle.
Citric acid cycle is the single best answer because it produces 2 CO2s for every turn of the cycle. The citric acid cycle produces CO2, and the oxidation of pyruvate to acetyl-CoA produces CO2. Just, that's self-is question. I'm not going to test you on that, but just to make the link between self-is and A&P. Okay, so CO2 is produced by the metabolically active tissues. What is this process of moving CO2 from the cells into the extracellular fluid around the metabolically active tissues? Diffusion. What is this movement, this arrow right here?
Diffusion.
Diffusion. And what are these arrows moving CO2 from here to here? Diffusion, right? Okay, diffusion. Enough diffusion. Well, no, we got some more. So CO2 diffuses from the cells that make it into the fluids, across membranes, into the blood plasma, into the red blood cell. The difference between O2 and CO2 is that a substantial amount of the CO2 that your body is carried as dissolved CO2. This is not trivial. This is substantial. Dissolved CO2 in the blood plasma. Remember, it's 40 millimeters of mercury. Okay. So now, some of that CO2 that has dissolved into the red blood cell stays as dissolved CO2. There it is right there, dissolved CO2. So CO2 is dissolved in the plasma and it's dissolved in the cytosol of red blood cells. Both of those are carrying substantial amounts of CO2 as dissolved gas. some of that CO2 actually binds to hemoglobin. And that was helping the hemoglobin let go of its oxygen, right? But a very small amount of it. So this is relatively small. Some of the CO2 is bound to hemoglobin in metabolically active tissues. What is this arrow here? So this is diffusion of CO2 from this point to this point. What is this arrow representing? CO2 plus water going to carbonic acid. What is this? Binding, it's this equilibrium right there. CO2 interacting chemically with water to become carbonic acid. What is Ca in this image right here? What is the name of that? Carbonic anhydrase is the enzyme that assists in this reaction by speeding it up 1,000,000-fold. So CO2 becomes carbonic acid. What is this arrow? So this shouldn't be a unidirectional arrow. This should be a bidirectional arrow because it's a equilibrium. What is this arrow, which also should be a bidirectional arrow? What do we call that arrow? Going from carbonic acid to bicarbonate and protons. Dissociation. So carbonic acid dissociates. But it also reverses, right? There's association of bicarbonated acid to make bicarbonate and protons to make carbonic acid. Okay, now here's an interesting thing. What is this process? And I'll give you a hint. There should be a circle drawn here. In fact, you should draw it on your handout. Draw a circle here that represents a protein. And this protein helps bicarbonate come out of the red blood cell, where it was produced by carbonic anhydrase and dissociation, and allows chloride to move in. This is a Sal-Phys level question. What is this protein?
Looking for a name or a type?
Type.
Is it anti-border?
It's an antiborder. It's an exchanger. It's the chloride bicarbonate exchanger. It's a protein that moves, because CO2 can diffuse, but can bicarbonate diffuse across this membrane? Can't happen, it's charged. It's energetically unfavorable, highly unfavorable. But we have evolved a mechanism to allow bicarbonate out of the red blood cell and into the blood plasma in exchange for chloride. The chloride bicarbonate exchanger, an antiborder. So now let's look at how CO2 is being carried in the blood. And I think I won't do this, I'll pick this up on Thursday. CO2 is carried as a dissolved gas in plasma and in cytosol of red blood cells. Does anyone have a sense of what percentage of this total volume is red blood cell? It's the hematocrit. What percentage of your blood is red blood cell in volume? What's a normal hematocrit? About 40%. So 40% of your blood volume is this, and 60% of your blood volume is this, and both of them are carrying CO2 as a dissolved molecule. In addition, your blood, red blood cells, and plasma are carrying CO2. This is key, don't close up yet. CO2 is being carried as bicarbonate ion. That is part of how your blood carries CO2. Converts it into bicarbonate and carries it in red blood cells, dissolved, and then exports it to a plasma where it is carried from the tissues that make it to the lungs where we're going to get rid of it on Thursday. Okay, so let's stop here. We'll pick this up on Thursday.